All right, everybody, we're going to go ahead and get started, I think. Please feel free to keep grabbing lunch and everything on your way in, but just in the interest of time, I think we're going to start. So first of all, thank you all so much for being here. I'm Alex Sockell. I am the segment lead for cancer genomics at PacBio. And I'm really excited to share with you why we think that accurate long-read sequencing is vital to understand cancer mechanisms so that we can ultimately do something about them, and also to share some really new data that we've just generated showing that with some of our new chemistry improvements, HiFi can now be applied to sample types that were not possible with long reads before. Oop. There we go. Cool. So first, just to explain what I mean by a HiFi read. So the way that this works is you start with high molecular weight, high quality DNA. We then ligate on some hairpin adapters called smart bells to create a circular molecule. And this is then bound to a polymerase and loaded into the bottom of a well called a ZMW, where the polymerase then reads around and around and around the circle many times. And because our error profile is completely random, we can then use those multiple passes to create a consensus. And it's this consensus read that we refer to as a HiFi read. So what this results in is reads that are both long, so these can be up to 25 kb in length, as well as accurate. So what we're showing here on this plot on the left is results from the Precision FDA Truth Challenge, comparing the accuracy of PacBio to Illumina. And so you can see that PacBio is as accurate or more so than Illumina, but they're up to 100 times longer. So I like this plot on the right because it's actually to scale, and it shows you how much more information you actually get from a HiFi read compared to a short read. The other thing that is really cool about our technology is that it includes direct detection of methylation. And so the way that this works is as the polymerase is incorporating fluorescent nucleotides, we are actually tracking the time that it takes for each nucleotide to be incorporated. And there are characteristic changes in the kinetics of the polymerase that occur when it encounters a modified base. And so as a result, when you sequence something on PacBio HiFi, you get A, C, T, and G, obviously, but you also get 5MC information, as well as we just announced today, 5HMC, and then also 6MA. Here we go. Cool. So what this means for cancer genomics is really the idea of the most comprehensive and multi-omic view of cancer that is available. So for DNA, what this means in green there is that you can look beyond just small variants like SNVs and indels to characterize structural variants, as well as to phase mutations into haplotypes to understand whether they are cis or in trans. For RNA, what this means is that we can look beyond just gene expression to actually see the complete repertoire of isoforms that are expressed by the cancer, as well as to discover novel fusions and characterize mutations that are actually expressed at the level of the RNA. And then lastly, for the epigenome, we're able to do, as I mentioned, the direct detection of methylation, and we can also characterize chromatin accessibility using a method called FiberSeq. Here we go. So why does this matter? Well, we can start with the central dogma of molecular biology. So this is something that we all know. DNA in the form of a gene gets transcribed to produce mRNA, which then gets translated into a protein. But if this appears oversimplified, it's because it is. In actuality, what this looks like is that the DNA is transcribed into multiple isoforms, and these mRNA isoforms get translated into multiple protein isoforms. And there's been data to show that these isoforms can be so different in structure and function that they essentially behave as if they were different genes. And this isn't just some genes. 95% of human genes that have more than one exon express multiple isoforms, with more than 7 isoforms per gene on average. But the challenge is that it's been shown that short-read sequencing misses 60 to 80% of this isoform diversity. And so what this means is that using short reads for transcriptome studies, we're missing a huge amount of variability, which, as you'll see in our customer talks in a minute, can really lead to incorrect biological conclusions, which is obviously not what we want. It's a similar story for structural variants on the DNA side. So the vast majority of genomic studies to date have really focused on variation in the form of SNVs and indels. But in terms of the amount of sequence that is affected, there's an order of magnitude more sequence affected by structural variants. And so that's what we're showing here with these circles. And the challenge, again, with short reads is that they really struggle to detect structural variants. So this is from a paper out of Mike Schatz's group showing the number of structural variants in a breast cancer patient sample detected by short reads in blue there compared to long reads in orange. And so you can really see that the vast majority of these events are only detectable with long read sequencing. To show a concrete example of why we would want comprehensive and multi-omic profiling, this is an example of an actual complex cancer case that we sequenced in collaboration with the University of Melbourne. This is actually from our poster at AACR last year. And this is an individual for whom short reads were not able to elucidate an oncogenic driver mechanism. And so there's a number of things we can see here. The first is that we have a large 29 kb deletion in tumor haplotype 1. And the reason that we know it is in this haplotype is because the long reads give us the ability to phase really easily into haplotypes. So it's really clear that this is a heterozygous event happening on one haplotype. But then with the overlay of methylation information, so that's what we're showing in red and blue here, red is hypermethylated, blue is hypomethylated. And what we see is that if you look at the promoter region right upstream of the BRCA2 gene here, you can see that we have hypermethylation of that promoter gene that's tumor specific. And so what this means is that this would be a complete loss of function of the BRCA2 tumor suppressor by two completely separate mechanisms, one genetic through structural variation and another through methylation here of the promoter, leading to a complete loss of function of that BRCA2 tumor suppressor. So this is really powerful, but there has historically been a limitation with HiFi sequencing that it required a lot of input DNA. The input DNA needed to be of high quality, but we have a couple of new advances that we've rolled out in the last couple of months that really significantly reduce this to make it much more possible to apply HiFi to a broad array of sample types. So the first is our SPRQ chemistry. So this we rolled out for our HiFi and will soon be on our Vega benchtop instrument as well. And what this does is it takes the input requirements from multiple micrograms down to the 500 or less nanograms as input that's required. And it increases the output per SMRT cell so that you can get a 40x human genome on one SMRT cell. The other thing that is really exciting, particularly for cancer research, is this new Ampli-Fi protocol. And so what this does is it actually enables HiFi sequencing with as little as 1 nanogram of DNA through a whole genome amplification-based method. And so what this means is that it actually enables the use of very challenging sample types with HiFi, including FFPE. So this is huge for us. This is actually the first time that we've been able to show that FFPE samples are able to be sequenced with HiFi. And so here we're showing an example of one of the first few samples that we sequenced. And so what you can see here from the read length distribution is that while we aren't getting reads that are up to 25 kb, we are getting reads that are significantly longer than we thought they might be. So here the mean read length is almost 3 kb. And what that means then is that because these fragments are shorter, we're actually sequencing more times around that circle. So this quality score is really high. So this is Q55, extremely high quality sequencing. And it enables us to detect a huge number of structural variants. One other thing that I'll point out, so while this initial run was at relatively lower coverage because the fragment length is shorter, we actually just got data off the sequencer yesterday showing that when you combine our Ampli-Fi protocol with our Connects concatenation method, you're actually able to get 40x genome coverage from a single SMRT cell from an FFPE sample. So really sufficient to do variant calling even for low frequency variants. So this is something that we're really excited about to really enable the sequencing of FFPE samples with HiFi. And so what this actually enables for FFPE samples when you're able to do this long-read sequencing is two main things here. So the first is the ability to phase mutations into haplotypes. So that's what we're showing here. This is the entire 200 kb EGFR gene. And what you can see here is that the reads are completely phased into the haplotypes in brown and purple there. And what that means, for example, in the case of a resistance mutation, if you have an individual who has 2 resistance mutations, you really need to know whether those mutations are happening starting on the same haplotype, meaning you still have a functional copy on the other, or if both copies are broken with mutations on both of the haplotypes. So this is really critical for understanding cis versus trans mutations and compound heterozygosity. The other thing that we see is that it really improves our ability to detect structural variants, particularly for structural variants in the sort of medium size range. So here what we're showing is a 300 base pair alloinsertion that we're able to not only call but phase into, in this case, an 8 kb haplotype block. And so what that then means is that we have a huge potential to uncover novel structural variants in these FFPE samples. And so if you have a cohort for whom you haven't been able to identify an actual causal biomarker, this is a case where you could apply HiFi sequencing to those archival samples and discover new variant types that then may explain the variation that you observe. Another historically challenging sample type for long-read sequencing has been cell-free DNA. And so the prevailing wisdom has been there, again, that cell-free DNA molecules are short. But actually, there was a paper out of Dennis Lowe's group a couple of years ago, which is what this plot is from, showing that if you apply HiFi sequencing to cell-free DNA, you actually see a relatively high percentage of long molecules. I think in this case, the longest molecule that they saw was 13 kb. And what that means, in a paper that just came out yesterday, last week, I'm sorry, they've developed a new model for methylation calling that they've then applied for cell-free DNA to predict cancer status. So you can see in the middle plot there that the methylation score for HCC patients is significantly higher than for the non-patients as well as for the HBV carriers. And also what they found is that applying this model, when they use only the molecules that are long, so have more than 7 CpG sites per molecule, you're able to see significantly better performance of the predictor using those long cfDNA molecules. And so something really exciting, this was very serendipitous this morning, is that we've actually announced that we've licensed this model from Dennis Lowe's group at the the University of Hong Kong. And what this means then is that not only are we able to call 5MC and 6MA, we are also now able to detect 5HMC, which is, I think people are knowing that this is a up-and-coming biomarker for things like liquid biopsy. So we're really excited about the possibilities of applying HiFi sequencing to these really complex sample types and to discover the types of novel findings that we're going to be able to be seeing in these new talks coming up in just a second. So we have our Arthur Dondi, who is from the computational biology group at ETH Zurich, and also Bingqing Zhao from the Snyder Lab at Stanford University, who are going to tell us about their work looking at both single cell and whole genome sequencing data from HiFi. Also, you should all stick around to the end because we have really cool prizes.
Okay. Hello, everyone. So welcome to the talk. And my name is Bingqing. So I'm a staff scientist from Mike Snyder's lab at Stanford. And today I want to talk about from colorectal polyps to cancer. So we really want to build a single-cell atlas for the regulatory landscape during the pre-cancer to cancer transitions. So a lot of you are cancer biologists, so experts here, and I do want to point again that colorectal cancer now is a very leading cause of cancer death in the United States and in the world, and both in women and men. And there is a very alarming trend of rising of early onset colorectal cancer. So there was an early onset colorectal cancer session this morning, which was really inspiring. And from this, we can see that in the past decades, that the incidence and mortality of colorectal cancer in elderly population, older than 65 and more, are steadily decreasing. I think that's due to the improved treatment and more pervasive screening. But alarmingly, that we are seeing a rising trend for early onset cancer, which happens in adults younger than 50 years old. So in Stanford, my lab was part of the H10 consortium, And then we built this colorectal precancer atlas, in which we focus on this group of patients with familial adenomatous polyposis, FAP. So FAP is a genetic disease that a lot of patients have a predisposition, a germline mutation in the APC gene, and this is autosomal dominant. So these patients will start to have a lot of polyps even from very young age. And at some point, they probably would have hundreds of polyps in their colon. And then if they are left untreated, they would almost always develop into colorectal cancer at some point. So the upper left panel shows you a real-world picture that we took during a colectomy, that because the patient was having too many polyps, so actually they sectored the whole colon out. So why we think the FAP samples are a very unique model for us to study this pre-cancer to cancer transition? Because in these patients, sometimes actually you can observe from normal to small, big, benign polyps to dysplastic polyp, even to cancer stage. So basically, you are seeing all the disease progression in one patient who has like the same germline mutation. So this is one paper that our lab published last year in Nature Cancer. And then what we saw here is that we profiled samples from 6 different FAP patients. And here we included samples types as normal mucosa, benign polyp, and the dysplastic polyp. So we performed mRNA sequencing, proteomics, lipidomics, metabolics. What we observed is that thousands of molecules change in every omic that we performed. And then they involve immune responses, cell proliferation, etc. What I want you to notice is that in this particular study, we actually don't have any cancer samples yet. But even with just polyps, we are seeing a lot of changes that's very cancer specific. So in an earlier publication from our lab in 2022, that we generated single-cell atlas maps for gene expression and chromatin accessibility, so single-cell RNA and single-cell attack libraries for over 500,000 cells. And this one we covered from normal to cancer stages, but we didn't really distinguish between benign polyp and dysplastic polyp. It's a complicated image and very busy. You don't have to look into every detail. But what I want to mention here is that from the immune cell populations, that you can already see a dramatic change in the polyp stage. And then we're also observing the cancer-associated fibroblast exclusively in the cancer sample, and also pre-cancer-associated fibroblast chromatin signatures in the cancer sample as well. So you may already notice that there are some gaps in these studies that we've already published. And what I want to do is really taking one step back to the genomic side and look into if extensive genomic alterations will rewire gene expression programs, shifting the cellular states in ways that would drive tumor progression. And the aim of the project is to perform a high-resolution multi-omic profiling of the colorectal polyp to cancer transition, identifying genomic alterations, RNA isoform dynamics, and the DNA methylation changes that are involved in tumor progression. And by high resolution here, I really mean in 2 ways. So one way is that we would distinguish between normal and then benign and dysplastic polyps. And another high resolution means that we will go on to single cell level as well as subcellular level. So here is my experimental design. So we actually have 4 stages of samples collected from the same donor: unaffected mucosa, benign polyp, dysplastic polyp, and cancer, which is adenocarcinoma in this case. And then for these samples, we would profile the genome using the long-read whole genome sequencing. The strength here is really to identify large-size structural variations. And then we want to look at the regulation side, looking at methylation this time. So we would get methylation together with our whole genome sequencing with the long read. And then I also performed the single cell methylation. For the transcriptomics that we're performing single cell RNA, both with the long read and short read. And then for the spatial RNA, we use the assay called stereo-seq. So that is one of the sequencing-based spatial transcriptomic assay, which would allow you to identify novel isoforms if sequenced deep enough. So why do we care about structural variation? So basically, this is a paper published last year on profiling over 2,000 colorectal cancer samples. And what the author shows here is that in almost every chromosome in our body that there might be a structural variation hotspot for colorectal cancer. And you don't need to go into the details on the left, but what I'm trying to show you is that there are a lot of structural variations that are much larger than 10 kb that would be very difficult for the short read sequencing to capture. And it's just so much easier with the long read when you have a direct readout. So now getting to our real-world data. So here I'm showing you the QC matrices from our long-read whole genome sequencing and methylation sequencing with the SPRQ reagent that's really fresh. So we got around 8 million HiFi reads per sample, and the data yield is around 90 to 100 gigabytes. And we have over 93% that has Q30 and over. So that means 99.9% accuracy. And the average actually is around like 99.95%. So we have around 30x coverage for this run. And the median polymerase read length are between around 70 to 80 kb. So this is the real HiFi read distribution from the run. For the 4 samples, you can see the HiFi median size is around like 11, 12 kb. And one first surprising and interesting finding that we had was that we actually observed whole genome doubling in our polyp samples rather than the cancer samples. A lot of cancer people would know that tetraploidy is quite common in cancer cell lines as well as many patient cancer samples. But I really couldn't find anyone claiming that seeing tetraploidy in the pre-cancer stage. And we're probably the first one really to see this. And I still don't understand why my cancer sample is not tetraploid, but that's something for later to explore. And then we look into the small variants that we have. So on the upper panel, I'm showing you the allele frequency of our small variants that you can see a steady increase as our disease progresses. So the benign polyp has around 10%, while the dysplastic polyp has about 20%, and the cancer actually has about 35% allele frequency of small variants. And also below, the pie chart shows you the composition of different types of small variants. First, you can already see that the number of small variants in each sample. So they rise from 1,600 to 8,000 to 17,000 in the cancer. And another thing to notice is that the percentage of deletions and insertions steadily increase from the benign polyp all the way to cancer. So another thing we look at is the mutational signatures. So what we have here is the single base substitution patterns, or SBS signatures. So in the benign polyp that you can see the most SNVs that we have are attributed to SPS 5 and 1. So these are 2 signatures that's common in the colon epithelial development, and they are to be seen in healthy people as well. And then in the dysplastic polyp, we're seeing a new signature coming up, which is the SBS 18, and that is related to oxidase effects. But in the cancer, it gets much, much more complicated. So we have multiple groups, signatures coming up, and I believe that one of the signature, SBS 85, is related to lymphocytes, and then we have the SBS 6, which is actually related to defective DNA repair. So that's very significant for the cancer sample. So next, we want to see what's shared between our samples across the trajectory. So here we filtered our small variants for the non-pathogenic genes. And then you can see that in the benign polyp that we have very few disease-related genes with small variants. And then we have a bit more in the dysplastic polyp and even more in the cancer. Interestingly, there is one gene that overlaps. So guess what? Yeah, so it's our APC gene. So APC gene is very known to take second hit during the colorectal cancer projection. And then we have, our donor has a germline APC mutation where I put, let me see. I have a little circled out here. And then in our samples that we profiled, we actually observed a second hit in every of our sample, but they are all different. So in the benign polyp, we have a splice region variant, which is determined to be of low impact. And then in our dysplastic polyp, we have a gained stop in a very early sequence before our germline mutation, and that's to be an high impact. And in our cancer sample, we also have a deletion, one base pair deletion, which will cause a frame shift, and it's also of high impact. So that's really interesting to learn that even though these samples are taken from the same donor, from the same colectomy, but they actually kind of independently developed their second hit on the same gene. And then, even though we have no other overlap between our dysplastic polyp and the cancer sample, but when we map all these genes into the signaling pathways, we find that the most genes actually converge into these very significant cancer-related pathways like Wnt, TGF-beta, PI3K, MAPK, EGFR, etc. So that's really interesting to learn. And next, we look into the structural variations. In our sample, we didn't discover a huge number of structural variations, and we have none in our benign polyp. But we do have a very interesting case that I want to illustrate in our cancer sample. That's the gene FBXW7. So that's one gene that's known to have deletions or other mutations in colorectal cancer, which is already published. But here, what we really observe is a large deletion that starts at the intron 3. So the deletion size, if you look at the breakpoints, one side is here and the one side is here. So the deletion is really over 400 kb. And that's probably the largest that no one ever reported before. And this deletion spans half of the, or several exons and the promoter of our gene, and deleting one gene in the middle, and also intrudes into the next gene. So we would expect some kind of isoforms there. And finally, we look into the differential methylation. And interestingly, that we have none in our benign polyp, but we have a huge overlap. So out of 52 differentially methylated genes that in the dysplastic polyp, 51 overlaps with our cancer sample. So again, we map them into the pathways that we're interested in, and the middle column shows you that, the overlapping genes that could be correlated to these pathways, and this is the dysplastic only, and that's the cancer only genes. Again, also cancer only genes, again, they have a majority that could map to these pathways. So I'm highlighting the genes here that we have evidence that the methylation is actually correlated to colorectal cancer. So I think this has huge implications on the early detection of dysplastic polyp and cancer, especially after I heard this morning's session on the early onset cancer, colorectal cancer thing, that the resource of colonoscopy is really limited. So once we reduce the screening age, that it's under a lot of pressure. But if we would be able to screen and find these patients with really dysplastic polyps already existing and find these biomarkers, and there's a good chance that we could catch them before the cancer occurs, but without wasting too much resources on the colonoscopy. Okay, so next I want to use a few minutes moving to my single-cell RNA part. So for the single-cell RNA, what we did was that we take fresh frozen tissues of all these different stages, and then we did a nuclei preparation. So we basically extracted nuclei from these samples, and then we captured the nuclei, barcoded them with the 10x system. And after cDNA amplification, we split the sample into two portions. So one goes on with the 10x short-read assay, and one with the long-read kinetics assay from PacBio. Just to mention here that because we're working with nuclei, not with cells, so we also incorporated an exome enrichment to enrich for more exonic reads. And of course, the short-read sequencing was sequenced on an Illumina X and NovaSeq X, and then we sequenced the long-read on the Reveal with the SPRQ reagents. And the analysis were done with the Cell Ranger and Surat for the short-read, and the SmartLink and Surat for the long-read. So just briefly how the kinetics work. So basically, you have your full-length cDNA, which are flanked by the TSO and the poly-A. And then we kind of wrap 16 of these full-length cDNA into one array using the array PCR. And then we put the mass adapters on both ends to make it into a hairpin and later a circle. So it could be read like for the really long whole genome sequencing. And after the sequencing is done, so you have these HiFi reads that actually contain 16 of your cDNA, and then you break them down using these connecting primers, and then you remove the adapters and TSO sequences, and finally you would have your cell barcode, UMI, and your transcript read. And this could be going to outputs that can be read into the Surat. So just some QC matrix for this run. It's even better than what we got with our whole genome sequencing, that we got over 100, like to 110 GB data, and then we have around 10 million reads per sample. So the Q30 plus bases are over 96%. And what the S reads mean that they are the single cDNA reads that's from the HiFi reads. So in total, we have around 150 to 160 million S reads. And then over 90% of them are FNLC reads. So these are the full-length reads that are flanked by the TSO and your poly-A. And of course, after dupe, we still have a lot of reads, and we identified over 25,000 unique genes and over 100,000 unique transcripts. So here I'm showing you the size distribution of the HiFi reads and the S reads. So the HiFi reads are similar to what we have seen in the whole genome sequencing, around 10 to 12 kb. And after we break them into 16 S reads, that our S read averages around 600 to 800, which is typical for kinetics assays. And the interesting thing is when we compare the barcode rank plot or the knee plot, that we observe very similar patterns from the long read and the short read single cell RNA data. So if you still remember that we split the sample after cDNA amplification, so it actually means you have exactly the same set of cells in your long read and in your short read. it will provide a lot of advantages in analysis steps later. Now I want to talk a little bit about the isoforms, which is the strength of the long-read sequencing. So here you don't need to remember the colors, but I just want to point to the blue and orange colors would be more or less your standard isoforms that uses the standard splicing junctions. And then your green and the hot pink ones will be the new ones. So when we look into our samples in real world, that we are seeing a very interesting trend. So for the blue and for the orange, if you look at the percentages, they keep decreasing from your unaffected all the way to the cancer sample. And then for the green and for the hot pink part, they kept increasing from unaffected all the way to cancer, meaning that you're having more and more novel isoforms with new junctions as the disease progresses. Lastly, I'll just show a bit of the single-cell results. So we definitely observe a dynamic change in cell type populations during the disease progression. So you can see from the unaffected that the cell populations shifts all the way to the right to the cancer sample. So these samples were co-embedded and batch corrected, but you can still see very clear shifts of cell populations. And you see that even better here. So when we plot the cell population into percentages, that you can see first that cancer-associated fibroblast exists only in cancer sample. That's the very little red bar on the top. And we have an increasing cyclin TA. We have increasing stem cell population. And we are losing the best 4+ and mature enterocytes, which are the functional units in a normal epithelial. So to summarize that the genomic mutation landscape really becomes increasingly complex as the disease advances, which is expected, but we're seeing a lot more details now. And many cancer-related genes exhibit genomic and epigenomic alterations as early as in the dysplastic polyp stage. So the benign polyps really show minimal genomic changes, but when we look into the cancer-specific cell types, that they emerge exclusively in cancer samples, but the global shift in cell type composition already began in the benign polyp stage. Okay, so that's for my talk. I really want to thank the people in my lab, in the pathology department, and in the sequencing call at Stanford. And I want to thank for the tremendous support from the PacBio team as well. And thank you for being here.
So now I will, a bit in the continuity of what Bingqing Zhao presented, I will talk about genomic and transcriptomic variant detection through long-read single-cell RNA sequencing in ovarian cancer and PacBio long-reads. And the main problem we have with ovarian cancer is that it's a deep lower abdomen tumor that will develop with very aspecific symptoms, meaning that this tumor will grow unattended and will spread into metastasis at some point, which in 17% of the case means late diagnosis, and that makes it a very deadly disease. And now you probably know that cancer mainly is deadly because of treatment resistance in the late stage. And one of the mechanisms behind this is that cancer cells will develop, will divide, and they will accumulate either treatment-resistant or treatment-sensitive variants amongst ovarian. And then after first treatment, a few cells that are treatment-resistant will probably survive, and there will be a relapse, and the second or third treatment won't work anymore. So now those variants are biomarkers for us that we want to track them, and we want also to, well, detect them first of all. So which variants are we talking about here? We think first of the genomic variants, the SNVs, of course, gene fusions, but also copy number alterations. But we can also think of transcriptomic variants like gene expression or alternative splicing, for example. And last but not least, here we are interested also in the microenvironment composition. Here I will not talk about also proteomic variants or metagenomics. That's also interesting, but that was not the focus of our study here. But basically, we want to detect the variants I showed here, and the best way for this will be single-cell RNA sequencing, because if you look here at the top, at the biological truth, you have a gene with maybe a single nucleotide variant. This gene will be expressed in multiple mRNA isoforms, and those isoforms will also carry the SNV. And basically, through the mRNA isoforms, we can detect all the variants I showed before. I will show examples later. But now if we look at the technological point of view, usually what you think of when you think of single-cell RNA sequencing is short reads. How it works, in most cases, you will capture the cDNA through the 3' end. You will fragment your cDNA into short fragments, and you will sequence fragments only around the 3' end, some on the 5' end, but mainly it will be concentrated on the 3' end, which means that basically from this information you can get the gene expression, and from the gene expression you will be able to derive the microenvironment composition and the copy number alteration, but not really the alternative splicing or SNVs, except if it's on the 5', let's say. Now, with long-read sequencing, you don't fragment anymore the cDNA, you still capture it on the 3' ends. So you have mainly, you have some at least complete transcripts, which are enough to basically detect single nucleotide variants, and also alternative splicing and fusions, which was missing with the short-read sequencing. So now I will show you the results that we had. I will tell you about my cohort first, and how we first detected genomic biomarkers, transcriptomic biomarkers, and through those biomarkers and variants, we were able to infer clonality and predict clonal resistance. And I will tell you why this was not enough to help the clinicians to choose the treatment later. But let's see first our cohort. So because the long-read single-cell RNA sequencing was first introduced in 2018 when we started this project, we had the idea of doing a small cohort here to test the technology. So here we have 3 ovarian cancer patients, and we sequenced their omentum metastases, 1 each. And for 2 samples, we have 2 patients also, we have healthy tissue sample as normal, pseudo-normal, let's say. And those patients were also involved in a larger study called the tumor profiler study that involves also some single-cell whole genome sequencing, pharmacology. There is also cytoph and mass spectrometry. So a lot of tests were performed on those samples, which means that we also had some confirmation samples to test on. And basically, now let's talk about the genomic biomarkers that we found in those samples. And first, the long-sum workflow that I built to detect those biomarkers, I built a SnakeMake workflow that was first will detect if the cells are correctly typed and differentiate between cancer and non-cancer cells. Then it will call the SNVs and the fusions, and through the SNVs and fusion, it will be able to detect clones. And on the other side, on the bottom, it will call also copy number alteration and also derive the clonality from those 2 information. And hopefully, you will have similar information from both data methodologies. So first, the single nucleotide variants that we found. In our 3 patients, we found 822 somatic SNVs. And as you can see here, it's a lot of intronic SNVs, 75% of intronic SNVs. This is for 2 big reasons. First, because we also capture some introns. The introns are like around 10 times longer than the exons, so that's also why you will find more here. And also they are less protected, meaning that you will just have more intronic mutations overall in general. But now, because those SNVs were detected in single-cell RNA sequencing, which is not really something that is classical, we wanted to, and also the editors, wanted us to validate those results with DNA. And as I told you, in those same samples, we had matching single-cell whole genome sequencing. And here you have the validation of all the mutations that had sufficient coverage of the 822 we had before. So it's around 120, if I remember well. And we had 80% of somatic support, meaning that in 80% of the case, we found mutations only in cancer cells using single-cell WGS, while we found 10% of or 11% of germline support, meaning that those mutations were indeed found, but they were also found in non-cancer cells. And then in 9% of the case, we had no support for mutation for two possible causes. Either we had a false positive in RNA, or also it could be also a clonal mutation, because those are not the exact same site of the biopsies. So it could be that the mutation is simply not there in the DNA. So also we found one TP53 with high impact SNV in each patient, which is what you would expect also in ovarian cancer. And this TP53 mutation was validated both here in single cell WGS, but also using panel sequencing. Now, talking about fusion variants now, we found multiple fusion variants, but this is the most interesting and also the most expressed. This is a fusion composed on the 5' of IGF2BP2, the first 4 exons, and on the 3' of TESPA1, only the 3' UTR. And the first interesting aspect of diffusion is that it was very highly expressed. It was 10 times more expressed than the wild type IGF2BP2, and there was basically no TESPA1 expression on the wild type because it's immune gene that was not expressed in those cells. And as you can see on the right, this fusion is not only highly expressed, but it's very cancer specific, as you would expect. And one of the reasons why it's so highly expressed is because it benefits from the IGF2BP2 promoters, but also from the lack of 3' UTR regulation of TESPA1. So it basically benefits of both advantage, which makes for a very high expression. But this very high expression also caused something disturbing at first, which was the expression of TESPA1. So in short read, when we looked at the result, TESPA1 is the highest differentially expressed gene in this patient, in patient 2. While when we look at long reads, this is not the case at all. And the discrepancy between those two results is because the short reads, they only read through the 3' UTR, and basically they are not long enough to go through the fusion breakpoint. So all those reads that you see here that express TESPA1 in short reads, they're actually from the IGF2BP2 TESPA1 fusion. And this is a big false positive. It's good that we didn't do any experiments on TESPA1 looking at short reads. This was also, as I said, those patients were involved in the tumor profiler study, and on an independent sample, they found the exact same result with a high TESPA1 expression. And if it was in more than one patient, they would have investigated actually on it. So this is it for the variants that are genomic. Now we will look at the transcriptomic biomarkers that we found. And first, looking at the novel isoform characterization. So as Bingqing showed before, we have in orange here the known isoform, and then in green and purple, the novel isoforms. And on the bottom left, you can see that we found actually 30% of our total isoforms are actually novel isoforms, which is huge. It's around here, I think it was 68,000 new isoforms. And when we try to validate those looking at the 5' and 3', so we match those isoforms with known experimental databases of 5' and 3' peaks, we had actually a very high support for those novel isoforms, as high as the orange, so as high as the known ones. But more interestingly, you can see that those isoforms have a much higher support than the gen code all, the third one from the right, which is basically the complete GenCode database. And this is because basically the GenCode database was built using short reads and also microfluidics, so it's quite old. And a lot of the isoforms there are actually not complete. So in general, we find actually isoforms that are of better quality than what you will find in GenCode. And we've sent those isoforms to GenCode actually, and they validated 40,000 of them, which would add up to 15% of their database, at least at the time, which is quite huge. That just shows you how far we are from characterizing the whole transcriptome. Now, looking at an example where there is a difference between cancer and non-cancer cells in isoform expression, the most interesting example we found was IGF-1, insulin-like growth factor 1, which is a hormone stimulating cell growth. And here in non-cancer, you can see on the right that non-cancer use the first exon. It's called the class 1 isoform, and this isoform is endogenous one, while cancer cells, they actually express the second isoform, and it's class 2 isoform, which is a secreted isoform, and we found that they do this because basically they secrete the insulin growth factor 1 to other cells to promote their development. And here I will not show you it, but we also found that an interesting case of mesothelial to mesenchymal transition that is actually stimulated by this type of interactions. And now that we talked about our genomic and transcriptomic biomarkers, let's look at the clonality. So usually, how do you call clones using the gene expression? You basically use a UMAP, and you will find your clusters. Here on the top right, you have our cancer cluster, and Surratt found two cancer clusters, one in red and one in green, and different type of non-cancer clusters. And that was a first result. Then we wanted to compare, can we find similar result looking then at the genomic biomarkers? So using SNVs and fusions, are we able to classify the cells in the same way that we classify them using the gene expression? And as you can see here, in green and in red, we find actually also two cancer clusters that have each cluster-specific mutations that are only expressed either in the green one or in the red one. And when looking then at the correlation between the clusters we find using SNVs and fusion and genomic expression, you can see here that we actually have an almost perfect match, meaning that we have basically two clones that are matching, which is expected, but this is good because now that we know that the isoforms and the different isoforms and the different SNVs and fusions are all clonal specific, not all, but we have some clonal specific, then we can ask the question, are those clones susceptible to be either treatment resistant or treatment specific? And the answer to this question is, in one of the same clones, we found an ALDHA3A2, SNV that is cisplatin sensitive, and this SNV here is specific to subclone A, and in subclone B we found an CCAR2 SNV that is also specific. This one is PARP-I resistant, and we also found a KRT8 and KRT18 downregulation that will indicate cisplatin sensitivity. So if we combine all this information, that means that subclone A is cisplatin sensitive, and subclone B2, while it is PARP-I resistant. So for this patient, we will recommend to use cisplatin instead of PARP-I. Now, did we pass those recommendations to the clinicians? The answer is, well, no. Because basically, this study took basically 3 years. So, of course, it was way too long to be useful for the patient. But that's normal because it was a test study. But still, the objective of this type of study is to build pipelines that are able to find those results in a few hours, which is actually possible once you have the sequencing data, but in a few days for now. And while this is possible, some of the obstacles that we find is that most of the variants, either SNVs, fusions of our isoforms, they are just undocumented. So you can find, for example, the fusion that we have, it's super highly expressed, but then what does it mean in terms of cancer? It's very hard to predict, honestly. And while for this, it is hard to predict. For example, for SNVs, there are ways to predict it using, for example, CRISPR-Cas9 gene editing panels. So I think it will get better, but for now, that's one of the biggest problems. We have to do this part after, like, a lot of the annotation has to be done manually, and like for 2 of the mutations I found, For example, I found that they were they were actually potentially meaning that it's cisplatting resistant because I looked at the literature. This was not a result that was automatized, sadly. Now, on the technical level, another problem was the insufficient long-read single-cell RNA throughput. This was before Kinnex and also before Revio, so now I think it will be better. We would have, we had here for 500 cells per sample, we had around 12,000 reads, which is already okay, reads per cells, which is already okay, but you would still want around 20 to 30,000 like you do with short reads. And also another problem is the fact that the reads are not complete, even though, and this is not the PacBio problem here, it's more on the single-seq technology side. The problem is that most of the time the reads are basically not complete, and in bulk RNA you will have around 3,000 base pair length, mean length of your reads, while for single-cell sequencing it's around 800, 1,000. So that limits a bit the, transcriptome characterization, basically. And the last problem that we had was, of course, the cohort side, because it's only 3 patients, but that was a test study. So, and with this, I am done. Thanks a lot. And I think that we take questions.
Awesome. So we can take questions now, and then also while we're doing that, if you look under your chair, there are secret hidden tickets under there, under a couple of the chairs, to win either this very cool Lego set or this secret gift box. And so if it's not under your chair and there's an empty chair next to you, you can look at the ones next to you as well. And then just bring that up here, and we can trade it for either up here or at the booth, and we'll trade it in for a prize. And then we'll all stay up here. So if you have questions and you just want to come talk to us, we'll all be up here. And thank you all so much again for coming.